This is the first video on the immune system. So the big picture that I wanted you to get from the immune system is, first of all, that immunity is both innate and adaptive. And secondly, that immunity is facilitated by both cells and antibodies. And what I mean by both innate and adaptive is that there are some portions of your immune system that are functioning from day one when you're born. And there are others that begin to function upon exposure to new pathogens or new environmental conditions. And we'll discuss the difference between those as we move on in this and the other videos on the immune system. So as far as disease resistance goes, you have innate or non-specific immunity. Now, as I said, these are present at birth, and they provide defense mechanisms that don't specify a particular pathogen, but are more sort of general protection against a wide range of pathogens. Adaptive or specific immunity involves the activation of specific lymphocytes, and these are white blood cells that are in the lymphatic system, and these will combat a specific pathogen or other foreign substance. So the anatomical system that actually hosts the immune responses is more the lymphatic system than anything else. So what are some general characteristics of innate defenses? First of all, they are present at birth. Secondly, they do offer even a newborn immediate protection against a broad variety of pathogens and of foreign substances. And no matter which pathogen that you're encountering, these protective mechanisms will function the same way. So whether you encounter a fungus or a bacterium or a virus, you're going to see a lot of these protective mechanisms function in the same way. So there are two lines of defense. One is the skin and the mucous membranes, and the second is internal defenses, and we'll go over those one at a time. So the internal defenses are going to be inflammation, and inflammation is going to have both cellular and chemical components. So our first line of defense here is the skin, and the skin is made up of keratinized stratified squamous epithelium. So you may recall from A&P1 that the skin is stratified squamous epithelium, but there's a topmost layer called the stratum corneum that contains a large amount of keratin in dead cells. And the thing about keratin is that it's resistant to bacterial enzymes and toxins, so it really does provide a good barrier against bacteria. And the second thing is the sebum, or the oil that's released onto the surface of the skin, contains chemicals that are toxic to most bacteria. I realize that most of you probably think of oily skin as something that promotes bacterial growth, but it's actually just a few species of bacteria that are able to prosper in that environment. So the mucosa is your epithelial lining on surfaces that are not covered by skin. And on mucosal surfaces, you have a number of things that keep bacteria from growing. First of all, an acidic environment inhibits bacterial growth, and so you're going to see particularly the stomach mucosa having being acidic and having protein digesting enzymes, but virtually all of the mucosa surfaces do have an acidic environment. Secondly, saliva and tears, lacrimal fluid, contains lysozyme, and this is an enzyme that is antibacterial. The third thing is, particularly in the respiratory and the digestive tract, you have mucus that traps microorganisms. In the respiratory tract, the cilia will sweep out the mucus. And last but certainly not least, you can have a normal bacterial flora where you have a mucosal surface. And the best examples of this are probably the mouth and the vagina. So again, the mucosal surfaces, they usually have saliva and lacrimal fluid that contains lysozyme. Again, mucus traps microorganisms, and these trapped microorganisms get moved out to a point of disposal by cilia. And again, the normal bacteria flora, and I've got some of the normal oral bacteria pictured here on the right, they will create an environment that's hostile to pathogenic bacteria. Another really good example of this is also in the GI tract, where the normal bacterial flora will create an acidic environment that is hostile to pathogenic bacteria. In fact, you may have heard of one of the more common probiotic bacteria being lactobacillus, and they get named after the lactic acid that they actually produce. So probiotics can, in my personal experience, provide some protection against lower GI tract infections. So I had a really nasty bout with diverticulitis about 11 or 12 years ago, and once I found out about the effects of probiotic bacteria on the pathogenic infections, generally if I have any of the kind of pain that might indicate a new oncoming attack of diverticulitis, I'll take probiotic bacteria, a triple or quadruple dose of that, and that will usually clear it up in a day or so, because for these bacteria, the environment in which they live, whether it's the mouth, the GI tract, the vagina, that's their home. And I've described these bacteria as being like residents of Texas. They don't like immigrants very much. So they're going to do their best to get rid of any species that's not theirs that's invading. So they do create an environment that's hostile to the pathogenic bacteria. And most people actually consider the bacterial flora a working part of your immune system. Okay, so internal defenses are your second line of defense, and these are usually going to come into play after either a mucosal membrane or the skin has been breached. And so what we're going to do is we're going to talk about antimicrobial substances, and this first one, interferons, are more accurately described as an antiviral defense. And so what interferons do is they'll do a number of different things. When they're released by an infected cell, they're going to signal, first of all, they're going to cause the cell itself to cease its own protein transcription. So protein transcription will totally shut down in the infected cell. And if protein transcription is shutting down, then it means that that particular cell won't be reproducing the virus that has infected it. It won't be producing that virus within it. Now, it'll also signal neighboring uninfected cells to destroy a lot of the mRNA that's present in their cytoplasm and to reduce their own protein synthesis to try to make sure that they don't start producing viruses themselves. So the other thing that interferons will do is to signal other infected cells to undergo apoptosis, which is programmed cell death. And finally, interferons will also activate immune cells such as leukocytes. And what these leukocytes will do is they'll travel around looking for virally infected cells, and they'll kill them. So three things are going to happen here that can cause the death of the cell that's producing the interferons. Number one, shutting down protein transcription completely will kill a cell. Secondly, it can signal other cells to undergo apoptosis. Thirdly, it can activate immune cells that will kill virally infected cells. Okay, as far as antimicrobial substances go, they have what's called the complement system. And so what happens first is the complement proteins, you can see here, that they insert into the membrane of bacteria, and they'll actually create a pore as indicated by the green arrow. Secondly, when those pores are sort of punched into the bacteria, water and ions will enter, and this will cause the target cell to swell up and burst. So basically, they're opening up holes in the bacterial cell membranes, which causes water to flood in and causes the bacteria themselves to burst. There are also antimicrobial substances. The top molecule on the right is an iron-binding protein. And so basically, bacterial growth occurs faster when there's a higher amount of available iron. And what these proteins will do is to decrease the available iron in the bloodstream. Now, this won't kill any bacteria, but what it will do is slow their growth down for long enough, or slow their growth down enough for your immune system to be able to kill them. The second protein on the lower right is an antimicrobial protein. And so these antimicrobial and also antifungal proteins will actively kill bacteria and fungi. But the second thing that they'll also do is when they bind to the bacteria and fungi and kill them, at the same time they'll also attract phagocytes to basically eat the bacteria and the fungi that they've bound to. Okay, so speaking of phagocytes, phagocytes basically are macrophages, and these pretty much descend from monocytes. So you've got a couple of different types of macrophages, depending on whether they can move out of the tissue that they're located in or not. So you have free macrophages that are able to move out of the tissue that they're localized to. These, you can find them in the alveoli, and you can also find them in the epidermis. Okay, so now we're going to talk about a couple of different types of fixed macrophages. One is the Kupffer cells of the liver, and the second is the microglia of the brain. And so these are limited to their particular tissues, and neither one of them, I think, is particularly good at combating microbes, but they do at least have a little bit of antimicrobial activity. Now, if you want to find cells that have a much greater antimicrobial activity, well, there are neutrophils, which are present in the bloodstream, and these are sort of the first responders to any sort of injury. You also have eosinophils, and these are weakly phagocytic, but they're mostly important against parasitic worms. And then you have mast cells, which are very similar to basophils. Now, they produce histamine and heparin in granules, and what histamine and heparin are going to do, these are going to be the chemical mediators of the inflammation reaction, and we'll go into that in more detail in just a little bit. So, how is it that phagocytes actually do their job? There are a number of steps that they take, and so the first step is going to be chemotaxis. And in chemotaxis, this is when phagocytes literally follow chemical cues on the microbes. So, I mean, there's no other way besides chemical cues for these cells to detect microbes. Of course, they don't see or hear, so they have to rely on chemicals in order to follow the microbes that they're pursuing. Now, once they catch up with the microbes, step number 2 occurs, and that's adherence. And so this is when the cell membrane of the phagocyte literally sticks to the bacterium that they're pursuing. The third thing that happens is ingestion. And in ingestion, you're going to see the bacteria or the microbe enveloped in what's called a phagosome. So this is basically a vesicle that the phagocyte has pulled into itself. Now, the phagosome is going to get merged with a lysosome in the process of digestion. And so basically, you have usually about 60 different digestive enzymes in a lysosome, and they'll be fully capable of breaking down that particular microbe. So there's the lysosome, and this leads to killing, which basically is what happens when those digestive enzymes hit and eat up the microbe that's in the phagosome. So the last thing that you see is a residual body, which can contain some indigestible material, but actually most of the chemical components of the microbe get recycled for use in the phagocyte. Okay, so the next thing that we're going to talk about is a type of cell that I think is maybe the coolest name in anatomy and physiology, and that's a natural killer cell. So a natural killer cell will attack any body cell that displays foreign membrane proteins, and this can be a variety of types of cells. So they can be cancer cells, they can be virally infected cells, they can be cells from transplanted organs, but the key here is what's called an MHC antigen complex. So the diagram on the right displays how the MHC antigen complex actually works. So in the first step, you have a small percentage of the endogenous antigens produced by the cell. So basically, these are proteins that are produced by the cell as part of its normal activities. And so what the cell's going to do is it's going to produce this antigen and then digest it into peptide fragments. At the same time, so there's the digestion. At the same time, you get the synthesis of MHC1 molecules. And so you're digesting this antigen into fragments, and you're synthesizing MHC1 molecules, which are the little things that are in green. Now, in the next step of this process, you see the antigen bound to the MHC1 molecules. And so now it becomes this sort of package. And this package gets placed into a vesicle, which then is placed into the plasma membrane of the cell in question. And so basically, this is a cell's way of saying, I'm a normal cell doing normal activities. Here's the evidence that I'm producing normal protein. Here's a fragment of one of my normal proteins that's being produced, and I'm going to show it on my surface. So if a natural killer cell actually encounters a normal MHC1 protein, then it will actually not attack the cell. If it detects an abnormal MHC1 protein, then what it's going to do is to attack the cell. If there's no MHC1 protein at all, it's still going to attack the cell. So cells that aren't displaying normal proteins are going to be cancer cells, which have mutated proteins, virally infected cells, which are displaying portions of the virus actually on their surface. Cells from transplanted organs are going to have a different genetic code, therefore a different protein structure, and they're going to look foreign as well to natural killer cells. So that's when natural killer cells can go after transplanted organs, and that's when you can see organ rejection. Okay, so with natural killer cells, what happens is a number of different things. So I want you to look at the diagram to the right. And first of all, you have what's called an activating receptor. So that activating receptor is a part of the natural killer cell. And what it does is to bind to, and so there's the activating receptor at the bottom. So here's the activating ligand at the top and the activating ligand at the bottom. So basically, the target cell has a ligand that can be bound by the activating receptor of the natural killer cells. Now, once this binding happens, it's very important that the normal cell have a normal MHC1 protein on its surface, because what that can do is to bind to an inhibitory receptor. Now, when that normal MHC1 protein binds to the inhibitory receptor, how does the natural killer cell respond? It responds by canceling lysis of the normal cell, so the normal cell gets left alone. Now, if you contrast that with the cell below, your target cell here has no MHC1 protein on it, which means that your inhibitory receptor doesn't get activated, which means that lysis will occur and that natural killer cell will attack the target cell and kill it. So I like this example because it shows lack of an MHC molecule, complete lack of an MHC molecule. And that can happen with some cancer cells. So if the cancer cells try to escape this whole system by just not producing an MHC molecule, they're still going to get killed. If cancer cells present an abnormal MHC1 molecule, they're going to get killed. If the target cell is showing a viral protein attached to the MHC molecule, it's going to get killed. So natural killer cells are able to respond to a broad spectrum of abnormal cells. And that's the reason why they're considered to be part of the innate defenses, because they're nonspecific and because they will respond the same way to any abnormality in a cell, not just a specific abnormality in a cell. Okay, so natural killer cells will, as I mentioned, also kill transplanted cells because they don't have the self-MHC1 antigen. They have an MHC1 antigen that originally came from a different person. So, of course, they're going to treat it as foreign. Now, once a natural killer cell encounters a cell that it's going to kill, the way that it does it is by producing enzymes called perforins. And what these perforins will do is they'll punch holes in the plasma membrane of the cell. And so to perforate something means to punch holes in it, and that's what these perforins actually do. Other enzymes called granzymes pass through those holes, and they activate the enzymes within the target cell that leads to programmed cell death. So basically, the MHC1 antigen is like a cellular ID card, and if you don't show the right ID card, then you get killed or you get instructed to commit suicide. So things can be pretty rough at the cellular level. Okay, so the next thing that we're going to do is talk about the inflammatory response. And so Here, with this diagram, if you look at the top left of the diagram, you're going to see step number 1. So in step number 1, tissues that get damaged by some sort of penetrating injury are going to release histamines, and what histamines are going to do, if you look at at number 2 at the lower left, they cause capillaries to become more leaky, and what that's going to do is to allow phagocytes easier access into the area, but it'll also allow clotting factors into the wound. So these clotting factors can actually kind of wall off some of the injury and prevent further spread by any pathogens that have gotten in. And of course, easier access of phagocytes into the area means that you're going to have these bacteria or whatever pathogens have come in to be cleared up more easily. So if you look at step number 3 on the right, this says that the phagocytes engulf bacteria, but they also engulf cells that have been killed by the injuries and cellular debris from cells that have been damaged or killed by the injury or by the pathogens. And finally, in step 4, you have platelets that move out of the capillary that seal off the wounded area. And so again, the whole idea here is to try to localize the injury as much as possible and keep things from going systemic. Okay, so now we're going to take a look at how tissues actually respond to injury and infection. So the injury and infection can be caused by, or the injury can be caused by physical trauma, by intense heat, by irritating chemicals. And so as there are always going to be pathogens out there that are going to be looking to take advantage of any injury that you have. And what are the overall effects of this response? To prevent the spread of microbes, to dispose of cell debris, and to really kind of provide a setting for tissue repair. So let's take a look at the diagram that's on the right. So whenever you have a tissue injury, one thing that happens is that you get the release of leukocytosis-inducing factor. And what leukocytosis is an increased number of white blood cells in the bloodstream. Now, once these numbers increase, then they're going to get migration to the injured area. And what's going to happen here is a couple of different processes after this migration to the injured area. You're going to get what's called margination, which is where the leukocytes are clinging to the capillary walls, and you can see that happening in this animation. And you can also get diapedesis, which is when leukocytes will flatten out and actually pass through the capillary walls between the cells of the capillary, and this is what you can see happening here. So you produce more white blood cells. They're able to cling to capillary walls and kind of control where they go instead of being swept along like the red blood cells that you see in here. And then they can squeeze in between cells of the capillaries. They don't damage the capillaries in doing so, but once they get into that injured site, then you're going to see the phagocytosis of pathogens and dead tissue cells. Now, in the short term, this is done by neutrophils. In the long term, this is done by macrophages, most typically monocytes. Now, at this point, pus may form as a result of the buildup of these white blood cells. Finally, when the area is cleared of debris, then you'll get healing beginning. Okay, so leukocytosis-inducing factor, that's not the only thing that's released during a tissue injury. You're also going to get the release of chemical mediators, such as histamine, complement, kinins, and prostaglandins. And so this does a number of things, this release does a number of things. First of all, it directly causes pain. Secondly, it's going to attract neutrophils, monocytes, and lymphocytes to the area. And so this is that chemotaxis phenomenon that we were talking about earlier. So if you're attracting neutrophils and monocytes, you're going to get, if you look at the arrow, increased migration to the injured area, and then the rest of those consequences happen. Now, the next thing that, or something else that happens at the same time, is an increased capillary permeability. And so these capillaries are going to leak fluid, and this is called exudate formation. As a result of this formation, you're going to get slowing of the blood flow, which allows for easier margination by leukocytes and easier diapedesis by leukocytes as well. Now, when the capillaries leak fluid into the area, they also leak clotting proteins. And so you're going to get this walling off process where the blood clot prevents injury to the surrounding area. In addition to this, the patch that's formed by fibrin actually forms a scaffold for repair. Now back up to our leakiness of our capillaries, you're also going to get additional protein-rich fluid in tissue spaces, and that's certainly going to cause both pain and swelling. But one thing that this can do on a larger scale is to give you a temporary limitation of joint movement. Now, if you have pretty severe swelling in a joint that you've injured, this swelling can actually temporarily reinforce the joint and maybe allow you to get back to a location where you can heal a little bit more safely. So you can kind of think of that almost as a boot, as a biological boot for the injury. Okay, back up to our release of chemical mediators. They also cause vasodilation of arterioles, and this causes a localized hyperemia, which is an increased blood flow to the area. Now, out of this, you're going to get an increased supply of oxygen and nutrients to the area, which is going to facilitate healing. You're also going to get redness, so this is the reason why inflammation makes an area look red. And you're also going to get, because of an increased blood flow to the area is going to get a little bit hotter, because if you'll recall, blood has a slightly higher temperature than other tissues. And so this slightly higher temperature increases the metabolic rate of cells, which increases healing. Okay, so again, the redness and the warmth comes from vasodilation, and the increased capillary permeability can give you edema. Now, the pain, as we've seen here, can come from the swelling, the edema, but it can also come from the chemicals that are released, the prostaglandins and the bradykinins. And so this is where you can get some loss of function in something like a joint. Okay, so, excuse me, the inflammatory mediators, which again, histamines and kinins, that result in vasodilation, that result in increased capillary permeability, this is going to increase the inflow to the injury site of exudate. And so, again, you're going to get some pain from the pressure of the exudate. You can also get localized pain from bacterial toxins. When this walling off process occurs, you can get pain from lack of nutrition to the area, and you can also get pain directly from prostaglandins and bradykinin. Now, what edema is going to do is it can act to dilute harmful substances like bacterial toxins. It can bring in nutrients and oxygen. It can allow the entry of clotting factors. And what this is going to do is eventually it's going to isolate the injured area and keep this injury localized instead of going systemic. Okay, so histamine is pictured on the right. It's produced by mast cells and basophils, and so this causes vasodilation and the increased permeability of blood vessels. And so both of these things put together are going to cause swelling. And so when is it that you usually take antihistamines? You take it to relieve sort of nasal congestion, so swelling of nasal passages in response to some sort of infection. Kinins are going to cause vasodilation, and so the diagram on the right is actually bradykinin. It's going to increase the permeability of blood vessels, and it's also going to serve as a chemotactic agent for phagocytes, so it's going to attract white blood cells to the area. Prostaglandins, which are produced by damaged cells, actually intensify the effects of those last two classes of chemicals, and they may also facilitate the immigration of phagocytes through capillaries. And another category of chemicals that we've got here are the leukotrienes, which, as with the others, can produce increased permeability of blood vessels and can serve as chemotactic agents for phagocytes. Now, the last thing that I've got written here is that it also causes them to stick better, and this is going to be good for a couple of different things. One is going to be for margination, but another thing is it's going to allow them to stick to microbes a little bit more easily. That's the adherence that we were talking about with the process of phagocytosis in an earlier slide. Okay, complement proteins, these will actually stimulate histamine release. They'll actually be chemotactic in nature, but they will also directly destroy bacteria. Okay, one of the last things that we're going to talk about is fever as an inflammatory response. And so your typical oral measurement is actually 98.2 or 36.2 C. When you start to climb above this, you have a fever. Now, body temperature actually does slightly differ from person to person, so your primary care physician pretty much has to establish what is a normal baseline for you. So fever is a systemic response, and the sort of set point for fever is set by the hypothalamus. So the hypothalamus is actually signaled to begin a fever by what's called chemicals that are called pyrogens, which are secreted by leukocytes. And pyrogen literally means to start a fire. So what fevers actually do is they cause zinc and iron to get stored in the spleen and the liver. It makes them unavailable to bacteria and really slows down their rate of cell division. So it's going to slow down the progression of this infection. But because it raises the metabolic rate, it can also speed up the repair of damaged tissue.